Hi everyone, my name is Kara Putri Gaisani and people usually call me Kara. And today I would like to discuss a topic about something that lies at the intersection of science, ethics, and also humanity. So while scientists ask what we can do, society must ask what should we do? So through this presentation, I will explain about what is human genetic, explore its scientific benefits, examine the ethical, and also discuss whether it should be allowed under strict conditions. So by the end of this presentation, I hope to provide a balanced perspective on this important and also a controversial issue. So first of all, let me introduce you guys to human genetic. So what is human genetic? Human genetic is a scientific process that could allow scientists to change, remove, or even add like a specific DNA sequences in human cells. But actually, gene editing is not only applicable to humans, but it is also eligible for plants, animals, and also other living lives. And the goal is often to correct the genetic mutations that usually could cause some diseases. So there are two types of human gene editing. The first one is somatic gene editing, and the second one is germline gene editing. So somatic gene editing usually targets a non-reproductive cells such as blood cells or even skin cells. The changes usually only affect the patient of being treated and are not passed on to the future generations. Because of this, somatic gene editing is generally considered more ethical and is already being used in lots of medical researches. For example, about how to treat a genetic disease such as sickle cell disease, and we will talk about this later. While on the other hand, germline gene editing usually targets embryos, eggs, or even sperm cells. Any genetic changes made at this stage will be inherited by future generations. So this type of gene editing is much more controversial because it raises some serious ethical, religious, and also safety concerns, including the lack of consent from future individuals. So in conclusion, while somatic gene editing is often supported for medical purposes, germline gene editing remains highly debated and is mostly restricted worldwide. And now, let's talk about science promises. One major benefit is the prevention of serious genetic disease. By correcting some faulty genes, scientists may be able to stop the inherited conditions before they cause a lifelong suffering. And then another important advantage is the improvement of quality of life. Gene editing, especially the somatic gene editing, can help patients to live a healthier lives without passing the genetic changes to future generations. And then human gene editing could also contribute to the advancement of science and medicine because it could allow some researchers to better understand genetic diseases and develop a more effective treatments. In addition, gene editing can be seen as an extension of medical care because it could be similar to like other accepted medical technologies such as gene therapy and also organ transplants. And then finally, by preventing a genetic disease early, gene editing has the potential to reduce the long-term healthcare burdens, both for families and even healthcare systems. So, despite of its scientific potential, human gene editing also raises a serious ethical concerns. One major issue is the safety risks and unintended consequences. Gene editing technologies may cause an unexpected genetic changes, and the long-term effects are still not fully understood. Another concern is the lack of consent. especially in germline gene editing, because the future generations cannot agree to genetic changes that will permanently affect their life. And then there is also a risk of designer babies, where gene editing could be used for non-medical purposes. It could be such as enhancing appearance or even intelligence, which may increase the social inequality. Additionally, many people raise the playing God arguments because believing that altering human genes can crosses the moral or religious boundaries by interfering with natural or divine creation. And then finally, there is a strong moral responsibility to future generations because since genetic changes can be inherited, society must consider whether it is ethical to make irreversible decisions without even knowing their long-term impact. So now I would like to discuss about the real-life example that I was talked about earlier, which is how to treat a sickle cell disease with CRISPR-Cas9. So, doctors can use a CRISPR-Cas9 gene editing on the patient's own bone marrow cells because bone marrow cells are like the source of red blood cells. And then, when the bone marrow cells already be edited, the edited cells will return back to the patient's own body. And then the treatment is used to correct the genetic problem that caused a sickle-shaped blood cells disease. So the real outcomes are patients who received their therapy usually stopped experiencing a severe pain crisis, and many no longer required a regular blood transfusions. And then in recent clinical trials, patients could show a dramatic improvement in quality of life. So, in conclusion, human gene editing presents a both great promise and also some serious challenges. So, I would like to recommend about human gene editing should not be completely banned, but it must be strictly regulated, limited to medical purposes, and also guided by ethical responsibility. Ultimately, the debate over human gene editing reminds us that scientific progress must always be balanced with moral values and also respect for human dignity. So that's all from me. Thank you for listening.